Hello, and thank you for attending today's webinar, Shaping the Future of Microbial Development, Gene to GMP, presented by Fujifilm Biotechnologies and Fierce Biotech. I'm John, and I'll be your moderator today. Before we get started, just a few housekeeping notes. Additional resources are available in the handouts window on your screen. To learn more about our speaker today, just head to the speaker bio window. If you close one of these windows and need to reopen it, you can just click on its name in the top navigation bar. This webinar is also being recorded and it will be available on demand within 24 hours. And finally, there will be a Q&A session at the end of the webinar today. So if you have any questions for our speaker, you can enter them anytime. Just use the submit questions window on your screen. Okay, I'd like to introduce our speaker today, Steve Loftus, Director of Commercial Technical Lead for Biologics at Fujifilm Biotechnologies. Okay, Steve, over to you.
Thanks for the intro, John. So my name's Steve Loftus and I am the Commercial Technical Lead here at Fujifilm Biotechnologies. And I cover our biologic services. So that's everything in the mammalian and microbial space. And my role really is to support customers on their early conversations with Fujifilm Biotechnologies and to act as a broad SME for all things biologics. So today I'm going to talk a little bit about some of the streamlined development approaches we take for microbial biologics to get you from gene to GMP. So Fujifilm Biotechnologies are part of a broader Fujifilm Life Sciences organization that aims to support clients from early drug discovery all the way through to a commercial launch. So within the life science space, we have divisions which supply media, they have specialty chemicals and reagents, and can also supply pluripotent stem cells as well. Over into the CDMO space, which is where Fujifilm Biotechnologies sits, we have the full range of modalities available for clients, from insect cells through to viral gene therapy. But we also are, a big kind of biologics manufacturer with mammalian cell culture and microbial fermentation. So we have this Partners for Life philosophy, which is something that we take from the early R&D phases that the clients enter into all the way through to commercial manufacture and supporting the patient long term. All of this is underpinned by our COJO-X manufacturing philosophy. So COJO-X is a standard of manufacturing, of facility design, which aims to transform the industry through three key pillars. Firstly, by putting our people first. So we are, a very, very people-driven organization where our employees, but also the patients that engage with the clients that come to us, they are the kind of the starting point for all of the journey that we take. We are a very kind of science and innovation focused company. So we have divisions which are focused solely on the next generation of bioprocessing. And finally, the whole kind of business is built on this unprecedented delivery of medicines to the client. And COJO-X allows us to have that unprecedented delivery by enabling us to manufacture on all of our sites in a standard way, using a standard facility design going forward. So moving on to our microbial experience. So we've been in the microbial bioprocessing business for quite a long time. So over 30 years, we have our own proprietary strains for yeast and E. coli. We've expressed over 150 individual proteins in those strains. We have 6 microbial commercial licenses across the network out of a total of 21 total licenses for E. coli, mammalian cells, Saccharomyces, Piscia pastoris, and we have some cell therapy commercial licenses as well. Our GMP track record is in excess of 2,500 GMP batches over the years, and at any one time we have approximately 50 microbial programs live within the global network at various different stages of development and manufacture. So The microbial sites are located primarily in two locations. So the Billingham site is kind of really one of our centers of excellence for microbial bioprocessing. And it's here that we do all of the strain development. We can do drug substance, clinical and commercial development. So we can do full de novo development for early clinical phases, or we can do process optimization for scaling up to commercial. We have fermenter scales from 150 to 5,000 liters. And we can also do some drug product development, as well as the ancillary services such as formulation development and advanced analytics. Over in our North Carolina site, it's broadly the same capabilities with the exception of the strain development. So we can do the clinical and commercial development and manufacturing at scales, again, up to 5,000 liters, but this is using a dual fermenter strategy in this case. And we can also do the drug product process development over there. And we also bring online in our Holly Springs site a clinical and commercial DP filling line, which can support our microbial business as well. But this is generally for some of the larger unit requirements up to 150,000 units. Over in our Hillerød site in Denmark, we have a commercial and clinical fill, which is what the North Carolina filling line will be based on. And again, this is for some larger fills up to 150,000 units. For smaller US-based clients, we have a DP filling line in Texas, which can handle up to about 10,000 units for generally clinical use. So now moving into the kind of the bulk of the talk. So in terms of early phase biotech and the customers that are going to be looking for a streamlined gene to GMP approach, what are they looking for when it comes to choosing a CDMO? So first and foremost, really, it's this technical expertise in multiple different modalities and the complexities of microbial-based biologics. A proven regulatory and quality track record is also very, very important, so that a customer knows that they're in safe hands when it comes to moving into the manufacturing phases, but also when it comes to filing their IND. This ability to scale the process from a very, very early phase one process through to commercial is something that really should be built into a program very early on. You don't really want to have to fully redevelop your process when you come to phase two supply or when you're moving into later phases. The early clinical phases can be quite dynamic. So having the speed and flexibility and responsiveness to respond to that dynamic changing landscape of an early clinical phase development program is also quite key. Transparent cost models. So customers really don't want to be presented with a very streamlined scope, but then later on down the line be then hit for upselling of several different services which weren't included in the original proposal. And it's difficult to manage budgets if you don't have that transparent cost model early on. And having a strong project management framework and a collaborative approach to be able to work with potentially quite small organizations and making them feel valued is also very, very key to long-term success. Some of their concerns, so budget overruns, this kind of comes back to the need for the transparent cost models. They don't really want hidden costs which are going to occur later on in the program. Delays and missed milestones can be, very troublesome, particularly when it comes to if they have clinical trials booked. A failed batch or extended PD can really significantly hit their clinical milestones and then later rounds of funding. The kind of the loss of control of their program and the deprioritization, they don't really want to feel that they've been bumped for a larger organization because they're quite a small partner. The risk of scale-up failures is very, very real. These processes can be complex. and sometimes they can be tricky to scale up. So having that expertise in developing the process with scale up in mind is quite key. There are, risks to early clinical phases when it comes to compliance and regulatory risks. That there is a very, very real chance of very, very early clinical failure, but having a kind of well-designed CMC package can support that regulatory filing. And as mentioned before, poor scalability, not just for that initial phase 1 batch, but also for later clinical stage, is something that needs to be taken into account. And really, we believe that Fujifilm Biotechnologies is a CDMO that can meet the needs of this type of early phase biotech client, while going quite a long way to mitigating the risks that they can see with this type of program. So as a full end-to-end CDMO, particularly for our microbial services as well, we can cover the full breadth of services from preclinical all the way through to regulatory approval and commercial launch. And you can see some of the tracks and packages of work that we have to support each of those clinical phases. But today, really, I'm just going to focus on the preclinical and early clinical phase development packages that we offer. So our Gene to GMP service is built on our Paveway Plus work package. So Paveway is our proprietary microbial strain development service, so it covers E. coli and Pichia pastoris, and it provides a fast and reliable, cost and efficient route to lead strain selection, which isn't just based on product titer, it's also based on product quality. And the product quality data generated during Paveway Plus is what is then used to drive the decision making and overall de-risk the later process development activities. So Paveway itself is E. coli expression system, which also encompasses some of our yeast strain development as well. For the E. coli system, it is antibiotic free. We have a number of innovative vectors and strains available to be able to direct the product to the right portion of the cell. So we can do insoluble expression, we can do a soluble cytoplasmic, and we can do periplasmic secretion as well. We have some standard, platform fermentation methods that we use, and these platform methods have been developed through many, many years of experience working with our selected host strains. We have, the ability to generate industry-leading titers. So titers of 20 gram a litre for E. coli are possible, and it's a well-proven system. We have a number of products which are entering into the later clinical phases, phase 3, and then entering into the kind of the validation phases soon. So it's a really well-proven expression system. And being able to be antibiotic free is something that is really beneficial when it comes to scaling up to larger scale manufacture. So you don't have to worry about the kind of the environmental and some of the clearance impacts of antibiotics. So in terms of the core workflow for our Paveway Plus, by the time you can get a titer readout in as little as 6 weeks, and this involves a couple of weeks of setup where we order the genes, and then we enter into a phase of cloning and using the Amber 250. So this is a high throughput 250 milliliter fermentation system that can run 24 fermentations in one go. So we have a standard approach to strain selection in that Amber, which allows us to run a number of different strains and a number of different vectors to really get the kind of the lead highest titer readout. And you can see in the chart on the top right, this shows you the kind of the titer increase over time, and it shows you the kind of the level of biomass that we get. And one of the key things about Paveway is that we have very, very tight control over expression pre-induction. So that allows us to increase biomass quite significantly before we trigger induction with IPTG, and that maximizes the amount of cells that are there to generate protein at the optimal phase within the fermentation. So once we have generated that lead strain, I mean, and here's some examples of the titer improvements that we can see. You can see that project number 1 there had very, very little expression before being dropped into paveway, and then this was a complex molecule, and we managed to get to just shy of 2 gram a liter after using the optimized vector and strains. Other titer improvements up to from 1 to 4 gram a liter. So we've got some significant titer improvements for really complex molecules here. But it's not just about titer; it's also about scalability. And paveway is a strain which can be scaled all the way through to 3,000 liter fermentation scale and indeed beyond. And you can see the chart there in the center. So this is plasma retention and OD600 plots from fermentations going from the amber 250, so 250 mils, all the way through to 3,000 liter working volume. And you can see a very, very, very tight cluster of data showing really, really quite impressive scale up all through the different scales that we operate. Plasma retention for paveway is also, very, very, really very high, with almost 100% plasma retention throughout. So it's all kind of well and good having a high titer, but there also needs to be some assurances that the product that you're making doesn't have any detrimental post-translational modifications. Some strains of E. coli, namely BL21, can add undesirable sugars and gluconolate the product of interest. So we need to make sure that we aren't just selecting on titer, that we are making the right quality of product as well. And to do that, we've generated a workflow which is based on a Tecan Evo liquid handling robot and robo columns. So we do a small scale extraction using the top 8 titer producing strains from the amber, and we then do a scale down extraction to then feed onto a resin screen. So our resin screen is focused on partial purification to be able to move on to some more advanced analytics. So we use a cation exchange and an anion exchange and a number of standard pHs and buffer conditions. In this particular example that you see, the product of interest bound to a anion exchange chromatography column. So these are small robo columns at 600 mil volume. We run a pseudo gradient to separate the product. And then once we have those fractions collected, we then run a capillary electrophoresis to then generate a product pool, which can be then taken on to some advanced analytics to get an idea of what the product quality is. So once we have identified the key resin, we can then the key binding conditions, we can then go on to do some scale-up chromatography, which is then used to generate larger amounts of material to do the analytics. And we can perform a standard UV method. We can do some STS page. We can do size exclusion, UPLC, capillary isoelectric focusing, and intact mass as well. All of this data is kind of compiled into a data package, which will allow us to kind of select the key producing strain. And the mass spectrum you can see in the center there is an example of some of the post-translational modifications that we can see. So in strains 1 and 2, so the top 2 spectra, you can see a prominent main product peak. However, in strains 3 and 4, there are 2 prominent peaks, and this is actually a reflection of gluconylation of the product which occurred in a BL21 strain for this molecule. This type of modification typically wouldn't be picked up till further down the line in later process development. So what we've managed to do is pick this modification up very, very early. So we immediately discount that strain, and we can focus on a strain which makes the correct material. So then we have high titer and we have correct PQ. And this then gets taken into the later process development. This workflow takes approximately 11 weeks. So the 6 weeks for the titer readout, and then onto the 11 weeks for the product analysis from the partial purification runs. We have also developed a workflow for insoluble proteins as well, which slots into that 11-week workflow. And this is based on an automated scale down solubilization screen in deep well plates. We test 12 solubilization buffers with a combination of different cheiotropes and denaturants and pH and buffers. And for this, we use an IB, an inclusion body slurry from a amber 250 scale down extraction. Once we've done the solubilization, we will then do a very kind of crude selection based on the amount of material that's still in solution. So using a high throughput A280, A550 screen. And then what is left in solution, we then do a CESDS screen to look at the level of solubilized monomeric protein. Once we have identified a number of different conditions, we then can move into a scale-up solubilization, which is then used to feed a high throughput refold screen. Again, it's based on the Tecan liquid handling robot, where we do a fractional factorial DOE design, which tests 190 unique buffers. And this is based on essentially past experience on the types of additives and excipients that have been used to successfully refold many different types of proteins. The high throughput screen generates what we term the refold index, and this is based on a absorbance readout, which looks at what has been successfully refolded versus what is kind of left in solution. We then use some DOE software to then pick out what would be the top 20 refold conditions to then take forward for some further screening. From this, we can select at least one refold condition, which can then feed into the partial purification in the capture screen that was previously mentioned. So this workflow then slots into the previous mentioned workflow, giving a product quality readout after 14 weeks. So then we've now selected a strain. We know the product quality is correct. We know if it's insoluble. We have some idea how to refold it. What we need to do now is then enter into some development beyond that Paveway Plus stage. So we have developed a number of streamlined workflows based on previous experience, and starting with chromatography development, again utilizing the Tecan high throughput liquid handling robot. So we've already identified the capture column. If that needs further refinement, that can be screened, and we can broaden the number of resins that we test on that. But essentially, each round of resin screening is broken down into 3 stages. The first of which is where we test up to 8 different resins using the Tecan. We do, again, the pseudo gradient. We collect the fractions and then use the CESDS to highlight where the product should be pulled. This will give us a lead resin based on where the optimal kind of product purity is for that capture step, or if it's the polishing step where the product is purest. We can then go on to a second stage, which is where we use a DOE to refine the loading conditions, the elution pH, and kind of build optimal separation between the product of interest and the less desirable impurities, whether that be product related or whether it be host related. So at the end of that, we have a lead chromatography condition and a number of lead resins, which are then taken into a scale-up run for final refinement, where we typically use columns of less than 100 mils and typically a 20 centimeter bed height. We do use some more advanced analytics at this point to then ensure that we are separating out any product-related impurities. We will refine the gradient. We will determine the binding capacity of the resin. And ultimately, if we can, move towards a step elution, which is much easier for manufacturing and avoids the needs to either do fraction collection and in-process testing. And it's simpler for manufacturing operations. And this is an approach that we take to both the capture, the polishing, and if there needs to be any final cleanup as well. So in terms of the analytical techniques that we use, so this is all with an eye on what will eventually be used for QC testing of the released drug substance. So starting with the very basic physical characteristics of pH, osmolality, conductivity, and the very basic safety testing like bioburden. We have platform methods that we use for these tests, which really just need verification with the product of interest. And actually the same goes for most of the analytical techniques that we employ. We have a base case off-the-shelf method that gets adjusted as we go through the development journey. Protein content now is almost exclusively using the solo VPE. We've moved away from the old manual dilution, so we only do the solo VPE, which is a more high throughput approach to it. Any identity and product purity and heterogeneity, we can do peptide mapping, and this can be chromatography based or it can be mass spec based. If it's chromatography based, it could be transferred into QC. We don't yet have the mass spec capability within QC. So this produces the kind of the peptide fingerprinting of the protein of interest and ensures that again there's no undesirable post-translational modifications. We of course need to quantify the process-related impurities and the host-related impurities, and again this is using generally off-the-shelf kits for host cell protein, for DNA, and for endotoxin. And we've recently begun to kind of consolidate all of these methods in-house now to avoid the need to outsource a lot of these tests to third parties. So we spent quite a lot of money recently in really upgrading our in-house QC capabilities to do all of this in-process testing. We do need to ensure that the product meets the purity standards set by the client. So most of our chromatography-based analytical techniques have now moved towards UPLC, which is a lot more high throughput friendly technique than the older HPLC methods. And also we've moved to capillary isoelectric focusing for charge heterogeneity testing. Finally, we have significant in-house capability to both transfer and fully develop cell-based and plate-based biological function assays. So we have some QC labs which have just been approved to run these tests and to develop these tests in-house as well. So one of the key things as well is to choose the right formulation buffer for your phase 1 biologic, and we have a staged approach to this as well. So starting with an initial pre-formulation personality assessment, and then moving through to some early stress testing where we'll kind of test for some accelerated stability, just to kind of get an early idea as to how stable this product is going to be. And for this, we use some advanced analytics such as DSC and DLS and a number of other techniques. Stage 2 is where we start to bring in a number of different buffers. We use different excipients. And it's at this point we start to narrow down what the formulation buffer might look like. And again, we use accelerated stability at this point to get some readout. The third stage is a predicted solubility screen, where we can look at the solubility at a range of different concentrations of buffer and excipient. And the final stage, when we've narrowed it down to a number of different key formulations, is we put those onto an accelerated and real-time stability study. And all of this runs in parallel to the process development, and we aim to get a formulation ready for the pilot scale run, or even sooner if the data is available. Finally, we also offer a huge range of protein characterization studies, and this can be quite important, particularly for some of the more complex microbial-based biologics. And it starts with some of the routine testing that we can do, whether that be peptide mapping, intact mass. If it is a glycosylated protein, we can release those glycans and do some glycan fingerprinting. For complex molecules, we can do a disulfide map to make sure that it's correctly disulfide bonded once we've got some purified material. And the key about this routine testing is that it needs to be right first time and it needs to be streamlined. So we need to have those platform approaches. We can do some extended protein characterization, where we can look at some complex charge variants. We can map the specific host cell proteins that are following the process through, give mass spec, subunit analysis if we have multi-subunit proteins. We can also look at sequence variants as well to make sure that there's no hidden mutations within the sequence of the product. And for this, you need a more specialized team of analysts. And for this, we have some subject matter experts who are trained and really well versed in each of these key techniques. And we can also work with external collaborators and the client themselves to develop new assays. And for this is where you kind of really need strong teamwork. So you've characterized your molecule, you've developed your process. What's the route to GMP for the molecule? So the formulation's been developed, that's now ready for the non-GMP pilot run. And at Fujifilm Biotechnologies, we try not to do an engineering run for our early clinical phase, because it's something that can increase the time and it can increase the cost. So we use a non-GMP pilot run in place of that engineering run. The methods are all qualified during the post pilot run activities alongside the GMP documentation prep. And these methods are qualified to a phase appropriate level. So they're appropriate for phase 1, and they're in a state when they're ready for further validation at later stages. The reference material, typically we take that from the non-GMP pilot run. So this is then used to test the drug substance when it's manufactured, and that's what forms the basis of the release, but also kind of is used to test the non-GMP stability studies as well, which kind of again run alongside the GMP prep and the GMP batch itself. What this ends up in is typically a 100-litre working volume GMP manufacture, and then the provision of released drug substance either to the client, or that can be taken into our internal drug product network, or we can work with a network of external drug product providers for the final released drug product. one of the things we've found with working with drug product providers externally is that it benefits both the client and the drug product house in that we retain the analytical methods for release of that drug product, and that avoids a costly transfer of methods out to the drug product filling house. All of the data generated during the development is CMC and IND submission ready, and we can support you during that preparation of the IND submission. So in terms of the timeline, so the expression studies and the PD take up the first 6 months. And at this point, this is where you'll get to the non-GMP pilot run. And then we can get to the QC released drug substance in approximately 11-12 months for a fairly standard microbial based product that we've experience working with before. We do have options to add on an additional chromatography or additional TFF step if the molecule is more complex and requires further process development. These additional stages may impact timelines, and we also have the ability to generate more bespoke analytical methods, which the client may require as well. So overall, the whole package aims to accelerate the strain and process development towards that GMP development, the GMP batch, by having a fully developed phase-appropriate fermentation and separations process. So this will be based on generally smallish scale equipment, so bucket centrifuges. It will be a minimum of 2 chromatography steps and a final TFF step. And it's 7 months to generating tox material and 11 months to generating released GMP drug substance. We have production scales up to 3,000 litre working volume, but typically for these first in human batches, it's at the 100 litre scale. If you are scaling up to 3,000 litres, then further work needs doing to generate a process which is suitable for that scale. So that's where we are right now. I'm now going to talk a little bit around, I guess, what the future may hold for the development of microbial-based biologics at Fujifilm. So the move towards platform processing for microbial-based biologics has been typically quite tricky because of the eclectic nature of microbial-based biologics and the lack of a defined purification tag. More often than not, regulators don't particularly like tags on biologics, and there's no readily available affinity for the whole range of molecules that you see within the microbial sphere. So what we did is we have a number of strategic partners within the universities within the UK and the US. So there was a particular UK university who were working on viruses for plants and the proteases associated with those. So we kind of reached out to them, and we kind of discussed a potential collaboration to develop a highly soluble protease enzyme which is readily expressed in E. coli, but also cleaves with very, very high specificity, but doesn't leave any residual amino acids on the protein of interest. So he did a little bit of a kind of deep dive into the types of proteins that we'd seen over the last few years. And generally, it's round about 75% are general recombinant proteins, and around about 25% are smaller proteins and large peptides. And what we found was that approximately 80% of the types of molecules that we see would be suitable for a tag and protease platform approach. So this covers, a real kind of broad spectrum of microbial-based biologics, and those that we found that weren't suitable tended to be some of the more complex molecules like VLPs which we've had expressed in microbial systems. So then that's led us to develop this kind of scarless protease platform. So we can design the molecule to have a fusion protein or a tag, which is then separated by a flexible linker which encompasses the cleavage sequence, and then you have the target protein. The final amino acid for our system needs to be any amino acid except proline. Taking a typical tag and protease approach, what you end up with is that terminal amino acid being retained by the target protein, which is something that generally isn't going to be accepted by a lot of regulators. Whereas with our scarless protease technology, we can release the target protein from the cleavage sequence and the tag, and then you can have a totally native terminal amino acid. So what we're moving to developing now is a couple of platform approaches to the development, and it's based on a histidine tag, a hist tag, and IMAC chromatography. So using the development approaches that I've mentioned previously, the fermentation and the separations are kind of fairly standard unit operations that we have work packages developed to define those. So we'll have the clarified product. This will go onto a standard approach to an IMAC column. We will then do the protease cleavage. This can either be done either in solution or it can be done on the column, depending on the efficiency of the cleavage of that particular product. We will then have a standard TFF, which will then get the product and the cleavage sequence, or any protease and any uncleaved product will be then put through another IMAC column via a flow-through mode. And because the protease also has a hist tag, this will bind to the metal ions on the IMAC column alongside the uncleaved material and any the protease tag. So what we are left with should be a relatively pure native target protein, which can then go into the product-specific purification steps using the high throughput methods that we've mentioned above. This also works for the insoluble route. So we have experience doing denatured IMAC columns on a number of different proteins, and we have found that we can do a denaturing IMAC to do the initial purification. We can then do the refold using a refold buffer that's been defined using the tagged protein early on, and then again entering into the standard unit operations of the flow through IMAC, and then on to the product specific purification steps. So this is a little glimpse into what the future will hold for microbial based biologics, and it starts to move to more platform unit operations, which will help standardize the development approaches and ultimately increase the speed and reduce the cost of getting to the clinic for early phase biotech. So finally, just to sum up, so the approaches that we take are kind of focused on speed and efficiency, where we have these accelerated timelines for molecules to get to tox in 6 months and up to released drug substance within 11 months. We have a very, very experienced team. We have decades of experience within the microbial sphere, and it's that experience which allows us to develop these streamlined approaches, which is built on the many different types of molecules that we've seen, and that's combined with the cutting edge high throughput technology that we use for the screening. We offer competitive pricing, which offers an affordable solution without compromising on product quality. And then we always ensure that we build in scalability very early on in the design of these processes. And finally, we have a very kind of collaborative approach. So we have dedicated project teams. So these teams come onto the project at the very start, and they stay with that project all the way through to the release of the drug substance and indeed beyond. So clients always know they're always talking to the same technical SMEs, the same project managers, when they are engaging in regular teleconferences. So thank you for listening, and I hope I've been able to provide some insights into some of the approaches that we take for microbial-based biologics. Microbial is a much smaller market than the mammalian market, and it needs these innovations to continue to push some of these more novel molecules to the clinic. So thank you.
All right, thank you, Steve. It's now time for our Q&A portion of the webinar today. Now, if you have not submitted a question yet, you can do so now. Just use that submit questions window on your screen. Okay, first question is this. What types of challenges are you seeing and hearing from customers to advance their project, and what else are you doing to support these?
So I think first and foremost, it's a very challenging environment for funding at the minute, particularly for some of the I guess the lesser represented modalities. So it's a landscape that is dominated by mammalian and antibodies, and what we've found recently is many of these smaller biotechs, which have some of the more novel molecules, they really struggle to secure enough funding to go all the way through from gene to GMP. So we have designed the packages of work to be somewhat stage gated. So we can break it at once we've identified a lead strain. We can then also break it after we've generated tox material, and then we can finally move on to the GMP. And then this is a way to kind of spread the cost of the development out so that the clients can then move from one funding milestone to another. So that's kind of one of the key things that we do. I suppose other challenges that they see is sometimes these molecules are quite complex. So aside from the streamlined approach, we can offer full bespoke development as well. So we can we've got a lot of experience with molecules such as VLPs, which we know the complexity of those. So we can do a lot more tailored development packages, which move further away from a platform approach, but they are well trodden paths that we know we can get those kind of novel and more complex molecules to GMP.
All right, thank you for that. Another question comes in: what mechanisms are in place to mitigate risk in reaching first-in-human manufacturing?
So we always like to build in a degree of quality by design very early on. So I think having well-characterized strains that we understand intimately means that the fermentation processes that we develop are based on a fairly narrow window that we know works and we know is scalable. We use the Amber 250 as the kind of the routine workhorse when we're developing the fermentation. But the problem with the Amber 250 is it has quite a wide operating range, and you can actually do an awful lot of things with the Amber 250 that you can't do in stainless steel vessels. So we purposely constrain the DOE around that initial development of the fermentation, because we know what our fermenters can handle when we go to scale up. So we're not trying to develop a process which ultimately won't be able to fit into a 3,000 litre fermenter, because the heat transfer is not optimized. Aside from that, again, we build scalability into all of the downstream that we're doing, and we make sure that as we scale the columns, that the columns aren't going to be too big for our assets, that the flow rates aren't going to exceed what we can do on our bioprocessing systems. And then when it comes to a 100-litre manufacture, we have matched equipment within our PD labs. So essentially, we don't need to do an engineering run, because we do a pilot run on the same equipment that we use in GMP manufacture. So this allows us to seamlessly transfer from pilot scale. We can write the documents which are based on protocols that we've generated for that pilot run, and then we can go straight into manufacture without the need to do the engineering run. And it all kind of because we've been doing it so long, it all kind of aims to de-risk that first manufacture, because ultimately this is development. There is risks in that very first manufacture that we don't really want to subject the client to, because we know there is funding relying on it. We know there is patients relying on it.
All right, thank you for that answer. Another question in, what is your approach past phase 1?
So I've mentioned, quality by design. We always have an eye on scale up. So when customers come back beyond phase 1 for perhaps their first larger scale manufacture, or even if they're coming towards phase 3 and then moving into process validation. So when it comes to scale up, the first scale up manufacture, we have packages of work which can be tailored towards defining the separations for scale up. So we'll utilize scale down disc stack centrifuges, which are kind of scale down models of our larger centrifuges, so we can define the primary separations in a more scale appropriate way. We can optimize the chromatography further to really kind of make it a commercial ready process, so we can eliminate as much as possible in process testing, have that inbuilt kind of process control very early on. We can start to identify critical quality attributes very early. And then as we start to move towards phase 3, we have in-house tools which can design FMEAs, we can have CQA mapping. And then ultimately this leads towards the assembly of a bespoke process characterization study. So we have dedicated equipment, we have dedicated scientists who can run process characterization studies. We have many years of doing process validation for microbial-based products. And we work with the client to design their characterization and validation package, which will get them towards BLA. And then ultimately we can support their commercial manufacture to meet their demand. And we have the flexibility as well for a number of different scales to meet, whatever their kind of their flexibility in the demand.
All right, thank you so much for that answer. And it looks like that's all the time we do have for today. Is there anything else you'd like to add as we wrap things up here?
No, I'd just like to thank everyone for listening, and if please reach out for any microbial development needs, but also for commercial manufacturing of microbial-based biologics. We have the capacity, we have the know-how to support you.
All right, thank you so much. And once again, that is all the time we do have for today. If we were not able to answer your question, we'll do our best to follow up after the webinar today. So as a reminder, a recording of this session will be available on demand within 24 hours. Once again, thank you for joining us today, and we look forward to seeing you next time.